So I'd like to thank the organizers for the opportunity to present our work, Enhanced Hereditary Cancer Screening through Scalable Parent of Origin Aware Genomic Analysis. My name is Kasmintan Schrader, and I'm Co-Medical Director of BC Cancer's Hereditary Cancer Program. I'd like to just acknowledge that we live and work on the traditional, ancestral, and unceded territories of the Coast Salish peoples from the Musqueam, Squamish, and Tsleil-Waututh nations, the Lekwungen-speaking people from the Esquimalt and Songhees First Nations, and the Stolo people from the Musqueam and Sumas First Nations. And we give thanks for the opportunity to live, work, and support care on these lands and beyond. I do have some disclosures that pertain to some of what I will speak about today. Just to level set on the technology that I'll be discussing, here is a technical video.
A key challenge in understanding genetic risk can be identifying which parent a pathogenic variant came from. Besides the X and Y chromosomes, every cell in the body has 22 pairs of chromosomes, one from each parent. To determine the source of gene variants, we can use a method called parent of origin aware genomic analysis, which looks at differences between the chromosomes using only a blood sample from the person with the variant. This method uses two main techniques, long-read sequencing and single-cell strand sequencing, also called strand-seq. Long-read sequencing is used to spot differences between the sequences of the two chromosomes inherited from each parent. It can also detect methylation of DNA at selected regions, called imprinted regions, that differ between the parental chromosomes, allowing for the identification of the parent of origin. By aligning multiple long-read DNA fragments to a reference genome, we can construct larger DNA segments with imprinted regions, revealing the parent of origin for each segment. However, if the variant in question is far from these imprinted regions, long-read sequencing alone does not provide enough information. That's where strand-seq comes in. This technique involves looking at live cells to track the forward and reverse strands that compose the DNA double helix. Cells are treated with a marker that labels new DNA strands. After the cell divides, the new DNA strands are destroyed, leaving the original strands, one from each parent. By comparing sequenced forward and reverse strands to a reference genome, it is possible to figure out which genetic variants are inherited together as a haplotype on the same chromosome, allowing us to tell the parental chromosomes apart. Typically, 20 to 50 cells are needed to get a complete picture. By combining parent of origin information from methylation patterns obtained through long-read sequencing with the haplotype details obtained with strand-seq, we can accurately determine the parental origin of each DNA segment. This combined approach allows us to map out the inheritance patterns from both parents across all chromosomes, giving a clear picture of how traits are passed down through the family and which side is at risk of carrying the pathogenic variant.
So just to recap, we are able to do this because we use 192 imprinted differentially methylated regions that are located across the genome with at least one on every autosome. And these are essentially detectable through Oxford Nanopore long-read sequencing, which is able to detect these 5-methylcytosines across the genomic landscape. So IDMRs, they are consistent differences in DNA methylation between maternally and paternally inherited alleles, and they're established in the gametes, and they either are established and escape the epigenetic reprogramming that happens after fertilization, or they're established after. And so all of these persist into adulthood. So with strand-seq creating the chromosome-length scaffolds, if you're able to phase these IDMRs to the chromosome-length haplotype, you're able to then determine the parent of origin of that haplotype in every variant along it with very high accuracy, as we showed in this paper in Cell Genomics for SNVs and indels. So what is the clinical utility in hereditary cancer? So up to 60% of individuals who inherit an SDHD pathogenic variant from their fathers will be at risk for pheochromocytomas and paragangliomas by age 60. This risk is not there if they have inherited it from their mother. And so there is a significant clinical utility in understanding this, and this is particularly important if parents are not available, because having it inherited from the paternal side means that there's lifelong monitoring from age 10 onwards with 2-yearly biochemistry, there's imaging, there's potential nuclear imaging, and there's direct and indirect burden to the patient and cost to the system. So what I'm showing you here is what the data looks like. These are Oxford Nanopore long reads, sequencing reads on chromosome 11, and this is showing a paternally inherited pathogenic variant in SDHD. And so the reads assigned to the maternal haplotype on the top show allele-specific methylation at imprinting center 2 at the Beckwith-Wiedemann syndrome locus on chromosome 11p at 1,5. And then on the paternal haplotype, which is on the bottom, you see the PV spanning reads where there is no methylation of the CPGs on that haplotype. And you see that only those, the paternal haplotype contains the variant reads. So this is the pedigree of the family, and what it shows is the squares are the males and the circles are the female assigned at birth. And the light blue show where we have applied the parent of origin aware genomic analysis, and with the little plus sign that indicates known molecular clinical findings, and the minus sign in the mother represents a known clinical molecular finding. And this is consistent with the parent of origin predictions that these are paternal in inheritance. And you see that in these 3 samples. So here's a quote from a patient where it's been applied with no unknown segregation of the pathogenic variant and following a paternal prediction. And so in this particular case, there was minimal cascade genetic testing in the family over 4 years, with tests only occurring on the maternal side. So parent of origin aware genomic analysis, although it's a research assay, it significantly and fundamentally alters the genetic risk assessment. And so instead of saying a BRCA1 mutation is what we classically would in clinic, say that there's a 50-50 chance it came from mom or dad, in this case, we would say that it's closer to 100% to the predicted parent of origin. In this case, it's the mother. And that risk is closer to 100%, with the relatives on the paternal side having a risk closer to the population or 0. And in this, of carrying the familial variant, and in this case, it also shows that the secondary relatives, so the uncles and the grandparents, their risk goes up to 50%. This is significant because in certain jurisdictions, that is actionable to be at a 50% risk of a mutation, even if the genetic status is not clarified. So if you're in BC, if you're a female and at 50% risk of a pathogenic variant in BRCA1, you would get access to MRI screening until your risk was clarified. So we chose to validate this in high-penetrant hereditary cancer susceptibility genes in real-world samples with known clinical molecular segregation. So they were in patients of differing age, sex, ancestry, and cancer status. And we chose genes with either parent of origin effects or where, if there was a pathogenic variant in a patient, that it might, the management might be dictated based on whether or not that variant segregated with certain types of disease in the family. So in 264 individuals with 265 pathogenic variants is the data that I'll present so far, where we have a good mix of self-reported ancestry. We've tried really hard to get a good representation across different ancestries to really try and understand the polymorphisms around the imprinted differentially methylated regions. And we've tried to also get a good sampling of sex assigned at birth, cancer history, and across different age ranges. Again, this requires live cells, so these were all individuals who gave a new blood sample. So we found that there was a high rate of assignment across different genes. So 85.6% of variants across 227 of 264 participants were able to have variants assigned. So as you can see, not all chromosomes are equal. And so as I showed you in the earlier slides, the IDMRs, although they're distributed across the genome, they're not evenly distributed. And so in chromosome 17 only has one IDMR used for prediction. And so if it's not present, then we are unable to do the assignment. However, when we are able to assign it, has a very high rate of concordance. So 98.2% of assigned variants were concordant, and that was 223 out of 227 participants. Or, sorry, variants in participants. So what I'm showing you here is a maternally inherited structural variant in MSH2. And this shows you that you can detect these large deletions with the Oxford Nanopore technology, and also that this is on, as you can see, the maternal haplotype, which is on the upper level of this, and as compared to the not contained within the paternal haplotype. So this is maternally inherited. And this is according to the IDMRs that we see. And here's an example of a paternally inherited structural variant in MSH2. Again, in this case, you can see the structural variants contained in the Oxford Nanopore reads. But in this case, the IDMRs indicate that this is paternal in origin on that lower haplotype. So there are factors that can limit parent of origin inference, and those can be errors in variant detection, like homopolymers, and that requires manual review or additional sequencing. There can also be rare phasing errors or sample swaps that can result in the wrong haplotype assignment of the variant or imprinted regions. And sample swap detection algorithms have been implemented, but there are rare phasing errors that can happen that might require additional nanopore strand-seq data. If there are runs of homozygosity at the known variant or the imprinted regions, this can impede our ability to assign parent of origin as well. And also, as I mentioned before, there might be a lack of allele-specific DNA methylation at known imprinted regions due to polymorphic imprinting, which can result in the inability to assign parent of origin. And this, what I'm showing you in these lower panels, is the IDMR located at chromosome 17. This is the only IDMR that we use. And on the left side, there's an example of no allele-specific methylation. And whereas on the right side, it's an example where you can see allele-specific methylation. So we are undertaking a clinical study at the moment where we are offering this research assay to newly diagnosed BRCA1 and BRCA2 pathogenic variant carriers. In this setting, we're offering it with an additional supported direct contact of their relatives and help using digital tools to try and increase rates of cascade genetic testing. The readouts of this should be available in 6 to 8 months, but we'll really try to determine if implementing parent of origin aware genomic analysis does help with cascade genetic testing compared to standard of care. We do anticipate the benefits to BC in 3 to 5 years with use of this technology to help direct offering cascade genetic testing to the side of the family that we deem to be most at risk through this parent of origin prediction. And this is using new methods of the patient-sanctioned direct contact or supported direct contact. We anticipate that we'll have the amount of genetic counseling and testing and will increase our mutation detection rates by doubling them and will result in a cost savings. So to conclude, genomic imprinting has significant clinical applications. IDMRs are polymorphic and will need characterization across different ancestries. POAga accurately infers parent of origin using only a blood sample, and there's a high rate of assignment and concordant prediction with clinical segregation in 264 cases where IDMRs are present. So POAga fundamentally changes the risk assessment, and that gives rise to the potential to improve cascade genetic testing efficiency, give more accurate a priori risk stratification in a family, and the potential to impact personal and clinical utility. And with that, I'd like to thank the patients and the families who participated in this research, my co-PIs Peter Lansdorp, Stephen Jones, talented trainees and now research associates Fahid Akbari and Lillian Cordova in particular, and all of our funders. Thank you.